So here we're going to go over the Michaelis-Menten plot for competitive inhibition. Now first, it's important to understand that competitive inhibitors are competing with the substrate for the active site of the enzyme. So if we take a look at the Michaelis-Menten plot, we can understand that on our x-axis we have our substrate concentration, and then on our y-axis we have our velocity, or our initial rates. Now, when we have a enzymatic reaction taking place with no inhibitor, no competitive inhibitor, this is what our plot's going to look like. But once we add in competitive inhibitors, we can see that there is a change to our plot. Now let's analyze what is this change and how is it affecting our plot. Now, up on top, we can see that we have this asymptote, and this asymptote designates Vmax. But what is Vmax? We have to understand that when we have an enzyme in a solution and we have substrate molecules surrounding that enzyme, at low substrate concentrations, we are going to have a little amount of substrate in our aqueous environment, and there is going to be a diffusion time of the substrate and the enzyme reaching each other. So the lower the substrate concentration is, the higher the diffusion time, because this enzyme is going to have to find the substrate, convert it into product, then it's going to go and find another substrate, convert it into product. But at high substrate concentrations, we are able to reduce that diffusion time, because by the time this enzyme converts this substrate into product, another substrate will reach that enzyme and it will convert it into product. And by the time this is done, another substrate will reach. So at high substrate concentrations, we are able to reduce the diffusion time of the enzyme and the substrate reaching each other. So at this point, we say that the enzyme has been saturated. So the enzyme is saturated with substrate. But it's really important to understand that the time it takes for the enzyme to recognize the substrate and convert it into product, that time period remains constant. And that is essentially our Vmax. Because at high enough substrate concentrations and our enzyme is saturated with substrate, it has substrate readily available, the time it takes for it to recognize it and convert it, that time is a constant. So at high substrate concentrations, we approach a maximum velocity. And that maximum velocity is just the recognition and conversion period. So that is our Vmax. And we can understand one key distinction of competitive inhibition. And that is when we increase the substrate concentration with our if we have a competitive inhibitor present and we increase the substrate concentration, our enzymes will be able to reach maximum velocity. Now, why is that? Remember, a competitive inhibitor is competing with the substrate for the active site. And if we increase the substrate concentration higher and higher, it will be more likely, the substrate will be more likely to reach the active site before the inhibitor does. And that is why at increasing substrate concentrations, we will reach Vmax, even if we have a competitive inhibitor. Now, this is a special feature of competitive inhibition. We do not see that in non-competitive and uncompetitive inhibition, because in non-competitive and uncompetitive inhibition, those inhibitors are binding to the allosteric site. Here, since the competitive inhibitor is binding to the active site, we can increase the substrate concentration, so it's more likely to reach the active site before the competitive inhibitor. Now, the second thing we need to make note of is our Km value. Now, Km is our substrate concentration when we are at 1/2 Vmax. So when we are at 1/2 Vmax. So if this is our Vmax, this would be half. Now at half, this is our substrate concentration for our normal enzyme, and then this is our substrate concentration for our competitive inhibitor, and these are our Km values. And we can make note that our Km is increasing. What does this mean? So remember, Km is 1/2 of our Vmax. This simply means we need more substrate to reach half of our maximum velocity. Whereas when we had no inhibitor present, we did not need as much substrate to reach half of our maximum velocity. And that makes sense because remember, the substrate is competing with the inhibitor. And in order to mitigate the effects of the inhibitor, we are going to need more substrate so that it is more likely to reach the active site of the enzyme before the inhibitor does. And that is why our Km increased and our Vmax stays the same because as we increase the substrate concentration. And that is essentially the Michaelis-Menten plot for competitive inhibition. We analyze the relationship between substrate concentration and initial rate. One final thing to make note of is that as we increase substrate concentration, our initial rate will also increase. So we see that proportionality in both of these plots because it's a proportional relationship. But as we increase substrate concentration, we will not continually increase initial rate. We will not see a linear relationship forever. At a certain point, we begin to plateau. Why are we plateauing? That goes back to our initial analogy of if we have an enzyme and we are saturated with substrate, we reach a maximum velocity, which is that constant time period of recognition and conversion. So at low substrate concentrations, we see that proportionality with initial rates, but then at high substrate concentrations, we begin to plateau and reach that Vmax, which is essentially when our enzyme is saturated with substrate.